Welcome back, everybody, to another deep dive. This time, we're going deep into the world of cannabinoids
Looking at the structure of these cannabinoid molecules.
and how those structures actually dictate their activity.
Exactly.
I'm very excited about this one.
Me too.
So we're talking about structure-activity relationship, right?
Yes, we are.
Or SAR.
SAR.
There you go.
And it's a very, very interesting concept in that even just small changes in the structure can have profound changes in the activity of the molecule.
So it's not just about whether a cannabinoid binds to a receptor, but how it binds
Exactly.
and what happens after, right?
And what happens after, that's a great point.
So this is where CB1 and CB2 come in.
That's right.
Right.
CB1 and CB2, the two most well-studied cannabinoid receptors.
Yeah.
And they have very different distributions in the body. Which is quite interesting. And when you look at the different molecules and how they bind, you can see that reflected in the effects that they have.
So like THC, for instance, we know that it's psychoactive. And that's because it's a strong agonist
Yes.
at CB1.
For CB1.
Yeah.
That's right.
Whereas CBD doesn't have that same psychoactivity.
Doesn't have the same psychoactive.
And we think that's because it binds differently.
Right. So it has a much lower affinity for CB1. And actually, some people think it might even be an antagonist at CB1.
Interesting.
But it does have activity at other receptors, like the serotonin receptor, 5-HT1A. So, it's really fascinating how just those subtle differences in structure can lead to these very different effects.
Yeah, so we're talking about not just THC and CBD here,
Right.
but also
All of the cannabinoids.
all the cannabinoids, endocannabinoids, phytocannabinoids, and even synthetic cannabinoids.
Right. All of them.
So can we break down the structure of these molecules a little bit more?
We can.
What are some of the key components that we should understand?
So they all share some common features. They all have a terpene group. They all have a cyclic ring system.
Okay.
And they all have various functional groups.
Okay, so those functional groups, those are like the attachments.
Those are the attachments.
That give each cannabinoid its own unique personality.
Exactly. And those functional groups can really affect how they interact with those receptors.
Yeah, so those functional groups, they're like the knobs and levers that
The knobs and levers.
determine how a cannabinoid interacts with the receptor.
Exactly. And a great example of that is the hydroxyl group. So if you have a hydroxyl group in a certain position, it might increase the molecule's affinity for CB2. But if you put, a methyl group there, it might actually prefer CB1.
Interesting.
Yeah, so really, really subtle changes can really affect those interactions.
So we're really talking about tweaking these molecular knobs and levers to fine-tune the effects of these cannabinoids. Is there a specific part of the cannabinoid molecule that's really crucial for binding?
Yeah, the side chain. So the side chain is often called the tail of the molecule.
Okay.
And that's the part that really interacts with the receptor. And the length and branching of that side chain can really affect how well it binds.
So does that mean a longer side chain leads to stronger binding?
Not necessarily.
Okay.
Sometimes a shorter side chain or one that's more branched can actually fit better into that binding pocket.
Oh, interesting.
Yeah.
So it's really about finding the perfect fit
Finding the perfect fit.
for that receptor.
Exactly.
It's like molecular Tetris or something.
Molecular Tetris. Yeah, I like that.
But it's not just about the shape, right?
Right.
There's more to it than that.
It's also about the specific arrangement of atoms that we call motifs.
Okay.
And those motifs kind of act like the teeth on a key
Okay.
that fit into the tumblers of a lock.
Gotcha.
And if you don't have the right motif, the cannabinoid just won't bind very well.
So you need both. You need that right shape.
Yes.
But you also need the right, motifs
Correct.
to really get in
You
on.
got to have both.
Yeah. So, what are some of the motifs that, scientists are really focusing on in their research?
Well, one important motif is one that involves a hydrogen bond between a hydroxyl group on the cannabinoid and a particular amino acid
Okay.
in the receptor.
So it's like a little molecular handshake?
Yeah,
Yeah.
little molecular handshake.
That really makes it stick.
Exactly.
So we've been talking about agonism and antagonism. Can we get even more specific than that?
They can.
How do researchers manipulate these structures
Yeah.
to really achieve those, nuanced effects?
So we can tweak those functional groups and we can tweak those motifs to fine-tune how a cannabinoid activates a receptor. So instead of just turning it on or off, we can create partial agonists.
Oh, interesting.
So they only partially activate the receptor.
So it's like a dimmer switch instead of
It's a dimmer switch.
just an on-off switch.
Yeah, exactly.
Gotcha. And then there's antagonism, where the cannabinoid actually blocks the receptor entirely. Right.
That's right.
Are researchers able to design cannabinoids that, say, only bind to CB1 or only bind to CB2?
They are.
Wow.
And that's actually a very, very exciting area of research. Because, for example, CB2 is mainly found in the immune system. So if you can develop a drug that only binds to CB2, you might be able to treat inflammation without having any of those psychoactive effects that come from activating CB1.
That's incredible.
Yeah.
We're really talking about designing cannabinoids that are like
Right.
precision tools.
Precision tools.
Right.
Yeah, exactly.
Targeting specific receptors to achieve the effects that we want.
Yeah.
Are there any other examples of, like, how scientists are manipulating these structures to really fine-tune cannabinoid activity?
There are, yeah. There's this really cool concept called biased agonism.
Oh, tell me about this.
So you know how we were talking about how cannabinoids can activate receptors? Well, it turns out that it's not just about turning it on or off. It's also about which signaling pathways get activated.
Oh, wow.
So, you know, when a cannabinoid binds to a receptor, it sets off a cascade of events inside the cell. And there are actually multiple pathways that can be activated.
Okay.
And with biased agonism, we can actually design cannabinoids that preferentially activate certain pathways over others.
So it's like choosing which dominoes to knock over in that, like, cellular chain reaction.
That's a great way to put it.
Wow.
And the implications of that are huge, because it means that we might be able to separate the beneficial effects of a cannabinoid from its side effects.
Oh, wow.
You know, so we might be able to develop drugs that are even more effective and even safer.
That's incredible. This is already getting so fascinating. We're just scratching the surface here
Scratching the surface.
of how intricate and really amazing this world of cannabinoid structure and activity really is.
There's so much more to uncover. So much more.
Okay, so let's get into some of these specific strategies that scientists are using to achieve those desired effects.
Okay, so
we can think about manipulating those functional groups. For example, if we introduce a hydroxyl group at a very specific position, it might enhance its affinity for this CB2 receptor.
Oh, so it's like we're adding a tiny magnet
Yeah.
to that molecule that draws it towards that CB2 receptor.
Yeah.
So then what about those motifs?
The motifs.
Those specific arrangements of atoms.
Yeah, so those motifs, those are super important because those are like the keys that fit into the locks of the receptors.
Okay.
So one very well-studied motif involves a hydrogen bond between a hydroxyl group on the cannabinoid and a very specific amino acid in the receptor's binding pocket.
So then if you were to disrupt that hydrogen bond, that might lead to antagonism?
It could lead to antagonism.
Or maybe if you modify that motif just slightly,
it might prefer a different receptor.
Yeah, it could shift its preference.
Yeah.
That's a great point.
Wow.
Yeah, it's like changing the teeth on a key to fit a different lock.
Gotcha.
Exactly.
This is incredible. So what about that tail of the molecule?
The tail, yeah.
The side chain.
The side chain.
How does manipulating that affect binding?
So the side chain is kind of like a rudder. You know, it guides the cannabinoid towards its target.
Okay.
And the length and branching of that side chain can really influence how it interacts with the receptor.
So if you have a longer side chain, does that necessarily mean that it binds more strongly?
Not necessarily. Sometimes a shorter side chain, or one that's more branched, can actually fit more snugly into that receptor's binding pocket.
Wow, so it's not just about size.
It's not just about size, no.
It's about finding that perfect fit.
Exactly.
Like a molecular jigsaw puzzle.
Molecular jigsaw puzzle. I like that.
And it's not always about creating the strongest binder either.
Right.
Right. Sometimes you want
to fine-tune the activity.
Yeah.
You want to maybe make it a biased agonist, or maybe you want to make it an antagonist to block the effects.
Wow. Okay, I'm starting to get a real sense of this intricate dance between structure and activity.
Yeah.
Let's bring this back to the real world.
Okay.
How are these insights being applied to develop potential therapies?
Yeah, so that's where the real excitement lies.
Yeah.
Researchers are now designing cannabinoids with incredible precision.
Okay.
You know, they're targeting specific receptors, and they're even targeting specific signaling pathways inside the cells.
Can you give me an example?
Yeah, absolutely. Let's take chronic pain, for example.
Okay.
You know, a lot of traditional painkillers, they come with a whole bunch of side effects. But imagine if we could develop a cannabinoid that only targeted the pain receptors that were involved without having all those nasty side effects.
That would be revolutionary.
It would be, yeah. And researchers are doing that.
They're actually doing that.
They are.
Wow.
They're meticulously tweaking the structure of these cannabinoids to create molecules that act like little molecular scalpels.
So instead of this sledgehammer approach,
Exactly.
we're really talking about like
a
precision strike.
A precision strike.
This is mind-blowing.
It is, and the applications are so vast. You know, we're not just talking about pain management. We're talking about therapies for inflammation, anxiety, neurodegenerative diseases, even cancer.
It feels like we're on the cusp of a whole new era of medicine here.
I think we are.
Where we can really harness the power of these molecules to treat such a wide range of conditions in ways that we never even thought possible before.
Yeah, and the most amazing thing is that we're just scratching the surface.
I was going to say.
There's so much more to learn.
There's so much more to explore.
Yeah.
Okay, I'm very eager to hear about these specific applications and what the future holds for this field. But first, we got to take a quick break.
Okay.
So that this all sink in.
Yeah.
And when we come back, we're going to explore some of the challenges and the future directions of this groundbreaking research. Okay, so we've talked about how scientists are tweaking those cannabinoid structures to create these really targeted therapies.
Yeah.
But bringing those therapies to the real world, I imagine that comes with its own set of challenges, right?
Oh, absolutely. There are a number of challenges, and one of the big ones is bioavailability.
Okay, so bioavailability, that's like
getting the cannabinoid to its target in the body in a way that's safe and effective.
So it's not enough to design the perfect molecule.
Yeah.
We've got to figure out how to deliver it.
Exactly. We need to make sure that it's absorbed properly, that it reaches the right place in the body, and that it sticks around long enough to do its job.
Okay, so that can get pretty complicated, I imagine.
It can.
Especially when we're talking about such a wide range of cannabinoids and all their potential uses.
Yeah, absolutely. There's a lot of research going on right now into different delivery methods.
Oh, okay.
For example, nanoparticles and liposomes.
I've heard about those.
Yeah, so these are tiny little capsules that can actually encapsulate the cannabinoid.
Oh, wow.
And then deliver it directly to the target cells or tissues.
So it's like we're sending a little molecular package right to its destination.
Exactly.
That's amazing. So we've talked about delivery. What about the fact that everybody responds to these things a little bit differently?
Right.
How does that factor into drug development?
Yeah, so individual variation is another huge challenge. You know, things like your genetics, your age, even your lifestyle can all affect how you metabolize and respond to cannabinoids.
So one size doesn't fit all.
One size definitely does not fit all.
When it comes to these things.
No. And that's why there's so much interest in personalized medicine these days.
Okay, so personalized cannabinoid therapy. That sounds
Yeah, so basically
wonderful.
Tailoring the treatment to the individual based on their unique biology.
So what kinds of advancements are really driving that personalized approach?
Well, genetic testing is a big one.
Okay.
So you can actually look at a person's genes and see if they have any specific variations that might affect how they respond to cannabinoids.
And that information can be used to guide dosing and even predict potential side effects.
Exactly. It's like having a roadmap for each individual.
It's pretty amazing. So we're just scratching the surface here, right?
Oh, yeah.
What else is coming down the pipeline in cannabinoid research? What are you excited about?
One of the things I'm really excited about is the development of allosteric modulators.
Okay, allosteric modulators. Break that down for me.
All right, so you know how we were talking about how cannabinoids bind to the receptors? Well, allosteric modulators, they bind to a different site on the receptor.
Oh, okay.
So instead of competing with the cannabinoid for that main binding site, they kind of sneak in through a side door. And they can actually change how the receptor responds to the cannabinoid.
So it's like they're turning the volume up or down?
Exactly. They can either enhance the effects of the cannabinoid or they can inhibit them.
So even more fine-tuning is possible?
Even more fine-tuning.
Wow. Well, this has been an incredible journey.
It has.
I feel like my mind is officially blown wide open.
Good.
We've explored how the shape of these molecules dictates how they behave in the body, how researchers are actually manipulating those structures to create some really incredible therapies. And we've even caught a glimpse of what that future of personalized cannabinoid therapy might look like.
Yeah, it's a really exciting time to be in this field.
It really is. And to our listeners, we hope this deep dive has left you just as excited as we are about the potential of cannabinoid research. Keep asking questions and keep learning, because there's a whole world of discovery out there.